Hello, this video is to help you with the chicken genetics gizmo. So let's take a look at it. Hopefully you've already gone through the slideshow talking about non-Mendelian inheritance, and hopefully you have seen that sometimes alleles don't just have a dominant and recessive form of expression. Sometimes they have something called incomplete dominance where they blend. So rather than one being dominant over the other, like brown over the white, instead they will blend to form a kind of intermediate color. So that's incomplete dominance. And these are some pretty puppies, and flowers can do the same thing where it's not just all red, but it's a blending of the two. And the other kind that I'll mention right now of inheritance is called codominance, where they'll both show up. It's not going to be a blending, but they'll both show up, and I'll show you a picture. So here's an example. A white cow and a brown cow, and they make, I think it's called a roan cow or something. But anyway, it's not a lighter color brown. It's actually some brown and some white. Chickens also do that. So here we have black and white, and then this is black feathers and white feathers. So this is an example of codominance where they'll both show up. Okay, so on that note, we're going to do this lab, and you can see this flower right here, same thing. It's expressing both alleles, the red color and the white color. It's not blending them, and it's not hiding one. So go ahead and answer those questions. We're going to go ahead and put a pure chicken to breed, pure red chicken, and pure white chicken. And we're going to click on Show Genotype, and you're going to, when I move it over, you'll see that the genotype for feathers is F big R, F big R for the red one, and F big W, F big W for the white one. You're going to answer those questions, okay? And then you're going to take a guess. What do you think that the offspring are going to look like when we breed them? You might already know the answer, but you can take a guess. And then we're going to go ahead and breed them, and voila, their offspring all have both white and red feathers. Okay, so describe the phenotype, what it looks like. So you're going to go ahead and do that. We're going to take 4 of them and put them in the holding cages, like we did the other one. Okay, and then we're going to clear this out. We're going to put 2 of them here. Let's not think too much about what's going on here, but we're going to breed these. Okay, several times. And each time we see sometimes they're white, sometimes they're red, sometimes they're white and red. Okay, and so you can answer that question. Now we're going to look at why this is happening, and we're going to use a Punnett Square to see if we can predict what's going on. So we'll go ahead and clear. We're going to bring these back. Okay. And before we do that, we're going to do a Punnett Square. So we're going to fill in the remaining squares. So this one is the first parent right here, okay? F big R, F big W. And then this parent is actually the same. So the alleles that are going to go in here are F big R and F big R. And the alleles that are going to go in here are going to be F big R and F big W. Now, if you don't know how to do the superscript, an easy way is just to copy this and paste it here. That way you don't have to bother with making those little W's and R's. So I'm going to copy it, and I'm going to paste it. Okay, so you can see that this one is going to be another red and white. This one is going to be all red. Okay, So I'm going to go ahead and do that for this square and this square. Okay. It's asking if the parents are homozygous or heterozygous. Well, these are the parents right here. And remember, homozygous means they have the same alleles, and heterozygous means they have different color alleles. So you have to look, are their alleles the same or different? And you're going to go ahead and explain that. And then you'll have your boxes all filled out, and you're going to write the possible genotypes. So here's one of the possible genotypes. You can just copy it and paste it. And then here's going to be the other one, the other one, the other one. Those are going to be the possible genotypes. And it's going to ask, are the offspring going to be homozygous or heterozygous? Okay, now we're going to calculate. So how many possible outcomes are there? You're going to look at these 4 boxes and see how many different outcomes are there. How many different genotypes are there? And then how many of them are going to be F big R, F big R, F big W, F big W, and F big R, F big W. Okay, and then you're going to put the probability. So you can just do, so let's say 2 of them are F big R, F big R. So you can put 2. And if one of them is this and one of them is this, then you'll put those numbers. And each box represents 25%. So if 2 boxes are going to be a certain genotype, then that will be 50%. If 1 box is a certain genotype, then that's going to be 25%. So you're going to go ahead and put, first you're going to just put how many of them have the genotype, and here you're going to put what percent. Okay, and then we're going to check it by actually breeding these. These are the 2 that you made the Punnett Square with. And you're going to breed it for till you get 100. So we'll show our statistics. And we'll go ahead and breed. Okay. And we got 1 red and 4 red and white. Okay, so that's our results of the cross. And the question is, did it match our prediction? Okay, and now we're going to breed it until a total of 100. Okay, and your results are going to be slightly different than mine. And here are some better percentages. And since it's out of 100, this is now 28%, 22%, and 50%. So you're going to compare it to these percentages that you got with your Punnett Square. And then that's about it. You'll go ahead and finish up any other questions. And hopefully this helps you. If you have any more questions, feel free to text me on Remind or send me an email.